Okay, we're going to talk about enzymes. Now, enzymes are biological catalysts, and any catalyst works by lowering the activation energy of the reaction. Right, they all work by lowering the activation energy. They don't get used up. That's the definition of a catalyst, which speeds up a reaction but doesn't get used up itself. Right, now the history of enzymes goes back to the 19th century, and let's just look at this reaction here, which is fermentation. Okay, now fermentation has been known for centuries. That's where you can turn glucose, which is C6H12O6, into ethanol and into carbon dioxide. And it's known that living organisms, yeast, are needed for that. Well, a scientist in the 19th century, he managed to kill the yeast and made a yeast extract. And he found that the yeast extract, when he put it in with the glucose, did the same job as the yeast. You still got ethanol and carbon dioxide. In actual fact, we know there's probably like greater than 10 enzymes involved in that. There's a whole load of different enzymes. But there's a basic idea. You have these molecules that have some kind of catalytic activity can make these reactions happen. Before that, there's some kind of magical idea that the yeast could do it just purely because it was a living thing. Okay, and that's where the word enzyme comes from. En means in, and zyme is to do with yeast. Zyme means yeast. So that's where the word comes from. Right, so for a while, people didn't know what the enzymes were, but towards the early 20th century, people began to realize that they were probably proteins. There's some controversy about that at first. Some people thought it was something that was bound to the protein. Eventually, people realized that enzymes were proteins. The fact that enzymes work at an optimum temperature of 37 degrees C, and proteins are denatured above that, that's obviously people realized something important going on there. Enzymes are very sensitive to changes in pH. Protein structure is very sensitive to pH. If you change the pH, you will denature protein. And so gradual realization that enzymes were probably proteins. Okay, now another thing about enzymes which we know about is that they are very specific. Okay, so let's think about two enzymes that we know about. These are both digestive enzymes. Okay, so the first one breaks down starch into maltose. That would be amylase. Say from the pancreas, pancreatic amylase, also produced in the salivary gland, and peptides. You've got this long peptide here, and it will break it into smaller peptides. Or the enzyme there would be trypsin would do that, which is another pancreatic enzyme. Actually, incidentally, it's worthwhile mentioning here that although both of these enzymes are involved in digestion, and most people's first exposure to enzymes is all to do with digestive enzymes, that is not their only role by any means at all. Every single chemical reaction in a cell, all of them are catalyzed by enzymes. All of the reactions involved in metabolism is all the reactions occurring in cells, are very slow by themselves, and they need to be catalyzed. So every single chemical reaction in a living cell is catalyzed, more or less anyway, by an enzyme. Right, just to say about specificity, starch cannot be broken down by trypsin, and we know peptides cannot be broken down by amylase. One enzyme seems to react on one sort of thing, which came to the idea of how do enzymes work. And I'm probably familiar with this. This is lock and key idea. So here we've got the enzyme in green, and this is a substrate, and that fits into the enzyme. And this bit where it fits, just colored in red, is called the active site. and that's where the catalysis occurs. Right, so this lock and key theory was that the substrate, explains the specificity of enzymes. The substrate needs to fall into the active site. Once it goes into the active site, the chemical reaction proceeds, and here are the two products. It's been broken down, the substrate's been broken down into two bits. This idea would explain the specificity of enzymes. It would also explain the fact that if you mess about with the temperature, you're going to lose the shape of the protein, so you're going to lose the shape of the active site. Same with pH. If you change pH, you're going to lose the shape of the active site, and so on. Okay, now that idea has been slightly modified by this new hypothesis, which is it's called, here is the induced fit hypothesis. Okay, so what have we got here? Well, we've got, this is this little hexagon is supposed to be glucose. You've got 6 carbons in glucose, and that one there is an oxygen. Okay, so there's 5 carbons in the ring, and this one here. This is actually ATP, adenosine triphosphate. Sorry, that is what I've drawn. There's ADP. I need to put another phosphate on the end. Okay, so that's ATP. And what this enzyme does is it transfers that red phosphate group on to the glucose, and that's, now we have got glucose 6-phosphate. Okay, that's a very important. That is one of the first reaction when glucose gets broken down, or it's oxidized into carbon dioxide and water in respiration. That's the very first reaction. So it's a pretty important reaction. And this enzyme is called hexokinase. This enzyme is called hexokinase. Hexo because it works on hexose sugars like glucose. And kinase, you'll see any enzyme that involves transferring phosphate groups is usually called a kinase. Right, so what happens is we've got glucose and ATP. I've drawn them here. Here's the active site of the enzyme. You can see the enzyme is here. It's got a little pocket for the glucose and another little pocket for the ATP to bind. And here we have got the substrate has bound. The substrates are bound. Right, and what happens as the substrate binds, the shape of the enzyme changes, okay, quite subtly. And then the reason why I've chosen this one, hexokinase, is because apparently the shape of the enzyme here changes quite a lot. So it alters its conformation. And this is a reflection of the fact that proteins, as we've seen before, we had a look at the structure of proteins. They're not big solid blobs like that green thing I've drawn there. They're made up of like alpha helix, beta sheets. There's like interactions between ionic groups. There are interactions between one hydrophobic part of the protein, another hydrophobic part, making it into molecular forces. So they are like they're kind of movable dynamic structures, and this is a reflection of that. So it's now thought that when the enzyme binds the substrate, you get a change in conformation, and that's called the induced fit hypothesis. Okay, and that's what is certainly thought to happen. Enzymes aren't rigid, immovable things. They are constantly adopting slightly different conformations. Okay, so that was the induced fit hypothesis, which is just a slight modification of the lock and key idea. Right, now let's look at some little reaction here. Okay, so what we're looking at there is sucrose. So this is a disaccharide. disaccharide, and it gets broken into two monosaccharides, glucose and fructose. And the enzyme that does this is called invertase. Okay, and invertase was an enzyme. Two very early experimenters, they're called Michaelis and Menton, and they did a lot of they did a lot of experiments looking at the rate of a chemical reaction and how it's affected by enzymes. So here's a graph of rate of reaction, and here they're changing the substrate concentration. And the substrate concentration in this case was sucrose. That's what they did all their experiments with, and they found they got this shaped graph. Okay, so they found that really you end up with you get a point there, and this is the maximum rate. And they hypothesized that what was going on here was when you get to this point, it doesn't matter if you increase the substrate concentration, the rate of reaction won't go any faster because all of the active sites of the enzymes are occupied. So they're all saturated with the substrate, and so you add more substrate, nothing more happens. The enzyme's working flat out as fast as it can, breaking down the substrate. Okay, and this early part of the graph here, well, here, if you add more substrate, the reaction does go faster because what's limiting it here is the amount of substrate, because not all the active sites are occupied. They did a lot of experiments with this enzyme invertase and a lot of mathematical analysis, much more complicated than that. And it all supported the idea that enzymes, the lock and key idea, where you had a site and the substrate fell into the active site and then got catalyzed, that everything pointed towards that happening. Okay, now let's have a look at enzyme inhibitors. So these are substances which can, so let's have a look at inhibitors here. All right, I'll rub this out. Let's have a bit of space. So enzyme inhibitors, they are substances which, if you add them to an enzyme catalyzed reaction, will slow it down. And they can fall into two categories. There are actually several different categories, but the two we need to know about at A level are competitive and non-competitive. Now, competitive, the way they work is these ones, they are the molecule, the inhibitor, is similar to the substrate. And so what they do is they kind of bind to the active site and they block the active site. So they fall into the little pocket and stop the real substrate going in and stop the enzyme working. Quite a few drugs work in this way. So if you have a look at this first reaction here, we've got serotonin, which is actually a neurotransmitter. So it's secreted by neurons, and that helps the nerve impulse cross the synapse. Low levels of serotonin are associated with depression. Okay, and serotonin is broken down to an inactive form there. So you can see that you lose an ammonia molecule, basically. Don't worry about that. And the enzyme that does that is called monoamine oxidase. And there's a class of drug which inhibit them. They're called monoamine oxidase inhibitors. And they are antidepressants, and they improve people's mood because they inhibit that enzyme that breaks down the serotonin. These drugs are similar in structure to serotonin themselves. They fall into the active site of the enzyme, stop the serotonin going in there, stop it being inactivated, and that improves the patient's mood. Okay, so that's one good example of a competitive inhibitor. Competitive because it's going for the same active site on the enzyme as the substrate. And here's another one which is probably even more familiar to you. Right, this enzyme is called cyclooxygenase. And it turns arachidonic acid, which is a fatty acid, reacts with oxygen and turns into this substance, prostaglandin. And what prostaglandin does, it promotes inflammation. Okay, so if you get if you like hurt your ankle or something and twist it and it swells up, it's prostaglandins being released which are causing your immune system to create that response. So if you can block inflammation, you can block the swelling and you can block pain. Right, so the inhibitors of this enzyme, competitive inhibitors, so these are molecules similar to the arachidonic acid, are ibuprofen and another one, paracetamol, and there's various other ones. Well, they're generally called non-steroidal anti-inflammatory drugs, and these are all similar to arachidonic acid in structure. They will bind into the active site of the enzyme, block it, and stop the production of prostaglandin and stop inflammation. Okay, so that's the competitive inhibitors. Now let's have a look at the non-competitive inhibitors, which is a little bit more complicated. We'll get rid of this. Okay. Right, non-competitive then. Well, so a good example of a non-competitive, well, what they do is if you have your enzyme, you'll have two sites. So, right, here's the round one is for the substrate. So there's the substrate's going to fit into the round hole. And that will bind to the active site, which is here. The inhibitor, which I'll draw in black here, is square. And that binds to this site here, binds in there, and that's a regulatory site. So what happens is the inhibitor binds into there. Once it goes in there, it changes the conformation of the enzyme, and it either slows down or stops the catalytic activity. And these are very important in metabolism, in controlling things. Sometimes you don't want enzymes to go on being active and doing their job all the time. You want them to stop. So let's have a little think about the glucose. Okay, so glucose in respiration, it's many different steps, and you end up with a substance called pyruvate. There's many more different steps, and then you end up getting carbon dioxide and water. Okay. Now, you don't want to be breaking glucose down into carbon dioxide and water unless it's actually needed for providing energy. And of course, if you've done this yet, you may not, but glucose, when it gets broken down to CO2, it ends up making ATP, which is kind of like the chemical, it's the energy currents of your cell. Okay, so it makes ATP. That's the whole point of doing it. So if you've got lots of ATP, you want to stop this process from occurring. Now in the middle here, there is a step here which I'll rub out, and there's the step here, and it ends up, you get fructose. So I'll draw fructose. fructose is a 6-carbon sugar, but it's got 1 carbon sticking down and 1 sticking up. And here you've got a phosphate group attached to it. So this is actually called fructose 6-phosphate. Just call it 6P. And it gets converted to fructose 1,6-phosphate. So you get you still got the phosphate there. You stuck another phosphate on there. So that's called fructose 1,6-bis bisphosphate. Now, the enzyme that that catalyzes this is called, it's a bit of a mouthful, it's called phosphofructokinase. And phosphofructokinase is the key enzyme which controls. You control that one, and you can stop the whole process of breaking down glucose into carbon dioxide and water. Now, the enzyme phosphofructokinase, if we draw it here, so it's the one. It's got 2 sites on it. It's got 1 site for the glucose 6-phosphate. So this is where the glucose 6-phosphate is going to bind. Sorry, fructose 6-phosphate. And you've got another site here, and this actually binds ATP. Right. So when you've got lots of ATP about, the ATP will bind to that regulatory site, and it will stop the enzyme from working. It is a non-competitive inhibitor. It's not competing with the active site. It's binding to a different site, to a regulatory site, and essentially switching the enzyme off when it's not needed. Okay, so I'll leave it there.